Hello students, welcome to EPG Pathshala. I am Dr. Jiten Vasist from Department of Biotechnology and Bioinformatics, JP University of Information Technology, Himachal Pradesh. Today I am going to talk about a module which entitled the axon and the resistance, which are myelinated and unmyelinated axons. This module is placed in paper Membrane Biophysics. Now coming to the axons, basically the axons are the nerve fibers which typically conduct electric impulses away from the neuron cell body and transmit information to different neurons, muscles and glands. The onset of an action potential at the axon terminus of a neuron initiates the process which is in turn propagates on adjacent cells and cause changes in the membrane potential of these adjacent cells. Present module primarily provides information of a typical axon, their structure-based classification, cause and mechanism of nerve impulse transmission through these axons. So for that matter, following learning objectives are prepared to achieve the said information. Number 1, to elucidate the structural and functional characteristics of an axon. Number 2, to classify the axon on the basis of their structural features. Number 3, to understand the effect of axon diameter and myelination on the conduction velocity. And last, to elucidate the mechanism of saltatory conductions through myelinated axons. A neuron cell has distinct regions like cell body, axon and dendrites, and each region has a characteristic functions. Basically, an axon and a dendrite are two protoplasmic protrusions that extrude from the cell body of a neuron. An axon is a long nerve fiber which typically conducts electrical impulses and transmit information to different neurons, muscles and glands. Although axons and dendrites are originated from the cell body, however, these can be distinguished from each other in several features which include shape, cell and their functions. In case of axons, they are usually longer, have a constant radius generally and transmit the information. While in case of dendrites, they are often smaller tapered as compared to axons. They are restricted to a small regions around a cell body and receive signals. Several neurons have no axon, and for that matter, the transmission of signals happens by their dendrites only. Now coming to the axon and its basic structure. In axon, the axolemma is the outer membrane of an axon in which exoplasm, which is a cytoplasm of axon, is enclosed. The branched end of an axon, we call it the axon terminal, closely adjoins the dendrite of another neuron, sometimes with the muscle or glands, and form a synaptic connection at junction, that's we call a synapse. Axons may be distinguished as unmyelinated and myelinated axons on the basis of the presence of a layer or sheet of a fatty insulating substance, that's we call a myelin. Myelinated axons are predominantly present in white matter, and myelin sheet has a contribution of approximately 50% of the dry weight of this white matter. Because of this, due to the presence of myelin sheet, a glistering white appearance in white matter is there, and it serves as a morphological distinction between white and gray matter. For the formation of myelin sheet, we require specialized glial cells which are responsible for formation of these myelin sheet around an axon. In central nervous system, these are oligodendrocytes which are responsible for making these sheets, while Schwann cells are required in peripheral nervous system for making these sheets. Then there another reason that's we call the axon initial segment or axon hillock. This is a thick unmyelinated region and contain a specialized protein complex. For example, voltage gated sodium channels. These are predominant or they are higher in this initial segment than the remainder of the axon or in the adjacent cell body. Their main function is as a site of producing action potential, and these are sites for initiation of action potential. The next portion is node of Renoir, which is present in the regular intervals in between the myelinated sheet. At the node of Renoir, there is a reduction in the diameter, and these node of Renoirs are responsible for making action potential gradient, and from this there is the action potential gradient is generated. Node of Renoir is responsible for saltatory conduction for fast propagation speed. The electrical currents are produced at each node of Renoir, and then these are conducted with little reduction to the next node which is in a line, where they remain strong enough to generate another action potential. The myelinated stretches in between the nodes get bypassed, and thus action potential effectively jump from one node to another node, resulting in a faster propagation. Now coming to the basic structure of myelination, that we have a myelin sheet. Myelin sheet, what is this basic components? Myelin sheet forms an electrically insulated layer around the axon and shield the axon from electrically charged ion which are present in the fluid surrounding the entire nervous system. Myelin is composed of alternate layers of lipids and proteins, and it has a high content of lipids. Approximately 70 to 85% are the lipids in the dry mass, and approximately 50 to 30% are the proteins. If you recall from our previous modules that this amount is different from other cell membranes, and this membrane has more amount of this one lipid as compared to the protein ratios. The primary lipid of a myelin sheet is a glycolipid, that's we call a galactocerebroside, but it also contains cholesterol, which is an essential constituent present in it. There are specific proteins like myelin basic proteins MBPs or myelin oligodendrocyte glycoproteins, and proteolipid proteins are several specific proteins which are present in the myelin. Myelin is made by different types of cells and varies in chemical composition and configuration. However, perform the same function, which is the insulating function for axons. As different types of glial cells are responsible for formation of myelin in central nervous system and in peripheral nervous system, the structure of myelin has some variations. In central nervous system, there are no specific lipids, but the contents of cerebrosides are directly proportional to the presence of myelin. Myelin basic protein and proteolipid proteins are abundant in central nervous system of most of the species. P0 glycoprotein and peripheral myelin protein 22 and P2 proteins are abundant in myelin of peripheral nervous system. The synthesis of myelin sheet is generally called as myelination, and in myelination, peripheral nervous system, the Schwann cells are responsible for synthesis of these cells, And in central nervous system, myelination of axons can be done with the help of oligodendrocytes. A fiber of say 80 Armstrong degree is surrounded by two protein layer that is around 15 Armstrong degree, in which both are separated by a lipid layer of approximately 50 Armstrong degrees. So what you will find that both molecule protein and lipids are surrounded by each other, and this forms a insulated layer that's called myelin, and thickness is then increased of this myelin by fusion of similar lipids and protein layers. Now coming to the another feature, that we have the action potentials which are required for transmission in axons or even for long transmissions. The signals are generated due to the change in the electric potential of a membrane or plasma membrane across the neuron. Electrical disturbances in one part spread to the other part is responsible for performing functions. However, these electrical disturbances become weaker when they are transmitted to a longer distance unless they get amplified. For that matter, for long communication, this passive spread is insufficient. Therefore, an active signaling has to be required, which is used by the neurons. In active signaling, electrical stimulus increased a threshold and triggers firing of electrical changes that spread along the neurons as automatic amplification. The term action potential is used for this traveling wave of electrical excitations. Axons having variable diameter from micrometer to millimeter are specific for conduction of the action potential. So the junction of axon and cell body, which we call axon hillock, is responsible for origination of an action potential. Then action potential has to be conducted down the axon terminal where it reaches the synapses, that is the small branches of axon that are connected with other cells. Axon potential is generated by a series of changes in voltage across the plasma membrane. A non-stimulated neuron is at its resting potential stage, that's near about 70 millivolt, and this has to be depolarized, and depolarization of neural membrane results in a peak action potential which is coming around say 30 millivolt. The rapid repolarization again is responsible for attaining resting value of the membrane. In axons, action potential moves down at a speed of up to 1 meter per second in non-myelinated, but the speed is insufficient to allow the complex movement of animals. For example, muscle contraction for walking or running would not be possible if it takes 1 second for moving an action potential from the spinal cord down the axon of a motor neuron to a leg muscle. It is the presence of myelin sheath which allows action potential to move at high speed or up to 100 meters per second that allow an action potential to travel the length 1 meter long axon and stimulate muscle in contract within 0.01 seconds. Now coming to the basic rules of conductance, resistance, which is also applied in axon physiology. Conductance through membrane act as an electrical circuit where internal and external environment act as a conductor and your lipid bilayer act as a capacitor. In the electric circuit, as you all know, according to Ohm's law, V or voltage is equal to multiplication of current and resistance. If resistance increases, the current will decrease. However, resistance will get increased with the length and decrease with the surface area. And if you recall, the surface area is a cylindrical formula for axons, and which is dependent upon diameter. It means what? The thickness of axons. Myelinated axons have larger diameter because of the myelinated sheet as compared to non-myelinated axons, and therefore their surface area will get increased, which has a higher or faster conductance of ion. Presence of channels act as a resistors as they allow ions to cross through membranes and hence reduces the concentration of ions also. In case of neuron transmission, two other factors also influences the propagation velocity of action potential. Number one, the length constant or the space constant, and number two, the time constant. The membrane voltage decreases both in time and in space and in the absence of any regenerative processes. So therefore a graded potential will decay as it travels away from the site of origin. Number one, if you see about this length constant, this decay may be described by the length constant in which corresponds to the distance where the graded potential has decreased to 37% of its original amplitude. The length constant can be defined with the formula that lambda is equal to under square of Rm divided by Ri plus R0, where Rm is the membrane resistance, Ri is the axial resistance, and R0 is the extracellular resistance, which is the force that impedes current flow through the extracellular fluid parallel to the membrane. In calculation, the effect of R0 are negligible. Therefore, the equation which can be typically said as the lambda is equal to square root of Rm divided by Ri. The length constant is dependent upon membrane resistance and the axial resistance. Membrane resistance is developed due to the leakiness of the membrane, and in that case the ions which cause the membrane potential changes move away from their source. They can move back into the membrane via other open channels, whereas the axial resistance is the internal resistance to current flow through the exoplasm, which is parallel to the membrane. The larger the diameter of the structure, the more is the opportunity there is for the ions to flow. Neurons with higher membrane resistance, which means few open channels and low axial resistance because of the larger diameter, will have a longer length constant. The large myelinated axons will have longer length constant than thinner unmyelinated axons, which means that the graded potential can travel over longer distances in large myelinated axons before dying it. Another factor is time constant, which is a function of membrane resistance Rm and the membrane capacitance, which is Cm, and which is defined as the combination of Rm plus Cm. Rm is the inverse of the membrane permeability. Therefore, the higher the permeability, the lower the resistance, and vice versa. Membranes can act as a capacitor and store charges. When a stimulus is developed, it takes time to charge up the membrane to its new value. Smaller time constant results in a rapid depolarization which affects the adjacent region. However, rapid effect of rapid depolarization to an adjacent region will bring the adjacent region to a threshold also sooner. Therefore, the smaller time constant will be required for rapid propagation velocity. Thus, the propagation velocity is directly proportional to the length constant and inversely proportional to the time constant. Now, after this, we'll just talk about the importance of myelination in axons. The importance of myelination can be observed in comparison when you compare between myelinated and non-myelinated neurons, their functions. If you see, the giant axons are present in squares and faster action potential transmission. The unmyelinated axons of squid, they are the giant axons having 500 micrometer diameter, which are 5000 times more energy, occupies 1500 times more space as compared to myelinated axon of frog having the diameter of 20 micrometer. So what we will find that non-myelinated neurons require a large area and require more energy to perform the same function when it can be done with the myelination in a small energy and a small region to be compacted. In myelinated neuron, conduction velocity is proportional to the diameter, whereas in unmyelinated neurons, it is proportional to the square root of the diameter. Myelinated coated axons have excellent cable properties, which result of faster depolarization through the membrane. This system or the property we call as a saltatory conductance, and this saltatory conduction is a property of myelinated neurons. Myelination have two major evolutionary sequences. One, the faster conduction of action potential, and second, conservation of metabolic energy, because active excitation occur in a confined region, that is at nodes of Rennewer. At the node of Rennewer, the axonal membrane is in direct contact with the extracellular fluid, which allows ions to cross this membrane. These nodes contain all the voltage gated sodium channels and all the sodium potassium pumps which sustain the ionic gradient in the axon. As a consequence, these myelin free nodes are responsible for inward movement of sodium ions that generate the action potential. The excess cytosolic positive ion at the node during the membrane depolarization associated spread passively through the axonal cytosol to the next node with very little loss, since they are unable to cross the myelinated axonal membrane. This caused rapid spread of depolarization from one node to the next node and permit the action potential to jump from one node to second node. Excitation of neurons by jumping of ions from nodes to nodes is called saltatory conduction, and is coming from the Latin word that's a saltare, means to jump. Now coming to the consequences which are happened because of degradation of myelin. Myelin is degraded from the axon, that's we call the axon demyelination. It's a result of degradation of oligodendroglia and Schwann cells by immune cells, leads to the multiple sclerosis. Patients which are suffering from this disease, it's a basically autoimmune disease, cannot walk through body like normal people. They used to walk instead of running because moving ion have slower velocity in demyelinated axons as compared to myelinated axons. Multiple sclerosis may cause disturbances in speech and may lead to sometimes paralysis. The name of the disease is sclerosis because once unmyelinated neuron fiber is formed, irregular dense scar tissue formed around it. These scar tissue are connective tissue such as fibroblasts that synthesize collagens. These collagen fibers are hard and hence resist the entry of sodium ions. This is the reason for slow movement of muscles. Now, as a whole, now we will summarize that what we have learned from this module. Number one, the axons of many neurons are encased by multiple layers of modified plasma membrane, that's we call the myelinate sheath. This membranous structure is elaborated by the adjacent supportive cells and facilitates the conduction of nerve impulses over long distances. Number 3, many glial cells are responsible for the formation of myelin sheath around the axon. Number 4, myelin formed by an individual glial cell is separate region from the next myelinated region by non-myelinated area of axonal membrane, that's we call the node of Renier. Conduction velocity of an action potential in non-myelinated neuron is proportional to the diameter of the axon, because thick axon can allow more and more ions to diffuse. Neurons with higher membrane resistance, because they contain few open channels, and lower axial resistance because of their large diameter, will have a longer length constant. The large myelinated axons will have a longer length constant than thinner unmyelinated axons, which means that gradient potential can travel over longer distance in large myelinated axons before dying out. The human brain is filled with comparatively small myelinated neurons. If human brains were not contain myelinated neurons, then they would have to increase axonal diameter about 10,000 times to achieve the conductance within 0.01 second as myelinated neurons do. Thus, vertebrate brains having compactly packed neurons could never have evolved without myelin. The myelin sheath and the node of Renier are responsible for saltatory conductions with the for fast propagation speed. The electrical currents are produced at each node of Renier, and then these are conducted with a little reduction to the next node in line, where they remain strong enough to generate another action potential. The myelinated stretches in between the nodes get bypassed, and thus action potentials effectively jumps from one node to another node, resulting in a faster propagation. Axon demyelination is a result of degradation of oligodendrocytes and Schwann cells by immune cells, lead to the disease that we call the multiple sclerosis. Patients suffering from this disease cannot move their body like normal people. They used to walk instead of running. That's end of this module. Thank you.